Welcome to our Genomics Lite, our April Genomics Lite, titled Genomic Cartography, Navigating with a Human Cell Atlas. And we're joined by Laure and Chloe, who we will get to hear from very, very shortly. Just for those that haven't been on a Genomics Lite before, I will quickly explain what it is. So our events are basically, it's an online webinar that you're in, you've made it, that's step one, which is great. And our events basically aim to help everybody to explore the science of genomics and what it means for our lives. So we ask that everybody participate respectfully. There'll be opportunities to put questions in the chat and have reflections and opportunities to put in your answers to polls. So we just ask that everybody sort of participates that in respectfully. There might be different views or experiences that come out in Q&As or stuff like that. I will be moderating the platform, and I'll be the person that hopefully that you won't experience any technical issues, but might be able to sort those out if you do. To interact with us, if you use the chat, that is the place that if you've got any questions, you can't quite hear us, maybe something's going a little bit wrong in terms of technical issues, something like that, you might have missed something, just put it in the chat. I will try and get back to you or direct you to the right place. If anyone would like subtitles with the event, there's a button that you should see which is called closed captions. If you click that, that will produce a nice transcript that goes alongside what we're talking about. And if you've got any questions that are specific to do with the content, if you put that in the Q&A button, we can get through to that. What I say is, if you've got any questions, put them in there straight away, and potentially we can either stop and talk about it if it's appropriate at that point, or we can get to it at the end. And to introduce you to where we are, my name is Sam. My role is a science engagement officer, and we are based at this place, which is the Wellcome Genome campus, on what is a very nice sunny day. I don't think it's that sunny anymore. I think we've had quite a bit of rain today, but that's where we're based. We're based in South Cambridgeshire, if any of you are watching from abroad. Yeah, we've got a few different organizations, such as the Wellcome Sanger Institute, EMBL EBI, and Wellcome Connecting Science, which is the organization I work for. The way that the session will go today is that we will have a talk from Laure and Chloe. Then, as I say, we might pause at points throughout the talk to go through any questions that anyone puts in, and we'll be wrapped up around about half five. The session will be recorded and will be available on our YouTube. So if for any reason your connection drops off or you need to leave at any point, there will be a recording, and we'll send that round after the event. So without further ado, we're going to start by having a little conversation about how Laure and Chloe got to where they are today. So starting off, Laure, why don't you tell us a little bit about your journey, essentially?
So yes, thank you, Sam. Good evening to everyone. Thank you for joining this webinar. It's our pleasure to share with you tonight our experience and career and day-to-day work at the Sanger Institute. So my journey started a while ago in science in 1995 in Paris, where I met this guy, Professor Gambardella at university. He was doing a lecture explaining how he found a way to treat people with extensive burn. And extensive burn can happen more often than we think. Just a house going in fire, or a car, or a nightclub, and you end, unfortunately, in the situation of this person in the hospital, losing all your skin. And without the protection, you cannot survive. We have just a few weeks to try save you. And he had found a way to, there's always a place in your body where the skin has been protected, for example, under your feet or under a belt. And if you take just 1 centimeter square, you have in this place the cells that are able to renew your skin all along your life. As you know, we renew our skin constantly, and those little things are stem cells, and I'm going to talk about them all along my presentation. And with the stem cells, he was putting it in a dish in his laboratory, he was able in a few weeks to build enough skin to save the person. That was amazing. I was fascinated by this, but unfortunately, those little cells did not have the power to redo the hair and to redo the sweat glands. So those people had a miserable life because they could not sweat and they did not have hair. So in his laboratory, we were intensively looking to break the secret of making hair follicles, the little things that are making the hair, and in an adult, because all those things are set up during your embryonic life. You cannot do them as an adult. So we were trying to understand how to make an adult making them or to make them in the laboratory. And I did my PhD on this. And we have other stem cells in our body that are able to renew the different places that we are constantly renewing. You probably know that you renew your red blood cells, you renew your white cells that are very important for the immune system. And this is thanks to the stem cells we have in the blood. And so after the skin, I went to the blood, and for that I came here in Cambridge because I also wanted to learn a bit more my English. So I knew I would keep the accent, but I was also hoping to have still a better English. And so I did 2 years with this Professor Anthony Green, and I learned about blood stem cells. But this stage, I preferred the skin. So I went back to Paris, but my lab had moved to Switzerland. So I spent 3 years in Lausanne, where I could ski. In the same time, I was doing some research. And so that was good. But unfortunately, my husband had a permanent position in Cambridge. And so it was complicated for our daughter. So I left this position and I embarked here again in Cambridge. And I decided at this stage to work on how the body developed. I wanted to understand how we set up all those organs. And I did 10 years of developmental biology, where I learned mostly how you develop the blood and the blood vessels and the immune system. And during that time, a lot of progress were made about those little stem cells, and some people were able to do plenty of other things in vitro. And it's like that I joined, because I was very excited by it, the laboratory of Professor Sanger in Cambridge at the Genomic Stem Cell Institute. He was able, from stem cells, to redo the cells of the heart that are the beating cells. So I can show you here the way those cells beat under the microscope. I think you can appreciate that it's very exciting when you can see that under the microscope. And during that period, someone in the state broke the secret of doing a hair follicle. The Professor Ms. Anifa at the Sanger Institute wanted to move this technique here at the Sanger Institute, and she advertised for a senior staff scientist that would do that. And I jumped on the position, and is where I could come here and develop now hair follicle in a dish. So that was my trouble.
Amazing, thank you. We lose you a little bit every now and then, so just for anybody watching it, we're slightly losing you at points through that, but hopefully people were able to get most of that. And then Chloe, let me try and, yeah, there we go, Chloe. Here we go. How about yourself?
So hi everyone, very excited to be here to talk about our science to you guys. So I started my research journey in France, where I enrolled for a bachelor and a master in pharmacy, because I was very interested in translational research, where we try to understand how the tissue work and how we can invent new drugs to cure diseases. After that, I had to do my pharmacy residency, and I chose to go in a psychiatric hospital, and also joined an oncology department in the same hospital, where I was responsible for constellation of medicine for patients to make sure that the cancer treatment, for example, were always on point and there was no error or mistakes in the way we were delivering them. From that, I did my first research internship in a different hospital in Paris, where I studied the fat tissue and its role in the development of HIV. And during this internship, I really became fascinated with immunology, which made me specialize with doing a master in immunology again in Paris. And from there, I joined another lab in another hospital in Paris to still study the HIV, but this time using a particular population of immune cells that are called dendritic cells to try and find a cure for HIV. And this was the study that led me to get a doctorate in pharmacy. And after that, in January 2022, I joined Mo's Hanifer lab here in Cambridge and started my PhD, where I decided to learn about a different population of immune cells that are called macrophages, that I will talk about in a moment, and their role during human skin development in utero.
Amazing. Thank you so much. And we can pick up some of these bits maybe when we go to the Q&A. I will stop sharing, and I know we've got a load of content to go through, so I will stop sharing and hand over to you both so that you're able to share your screen.
So hopefully the network will work.
Yep, we're seeing that all fine our end at the moment.
So this session is in fact part 2 of webinar of what's called the genomic cartography. And so I decided, because I was expecting that some people would join only tonight, I did a little summary of what was said in the first session. In the first session, our colleague working in the lab with us, Elena and Ferry, introduced the concept of the cell as the little brick of the body. Our body is made of little units that we call cell, and you just need to imagine a wall, a house, and the different bricks. So those different bricks, they do layer, like we can do layer and wall in a house. And when they accumulate, they do organ, and the organ are making your body. So, but how a cells know that it belongs to the stomach or to the eye or to the kidney? So, because every cell of our body has all the information, the genetic information in the DNA to do everything initially. But some of the cells, depending the signals they receive and a lot of things, they are focusing on a role. And to focus on this role, they look only at some part of the genetic information. They transcribe a part of it in another language that is called the mRNA. And this mRNA is going to give the function of the cell somehow about making some proteins that are going to do the machinery. So if we know exactly the mRNA of the cells, we know the function of the cells and exactly which part of the body it belongs to. And it's like that Elena and Ferry last time explained how we make an atlas of the human body. So today you just have to accept that we are able nowadays to do an atlas of the human body, and we are going to explain you what we could do thanks to this atlas. And we can do several things that are quite important in laboratory. We can, from this, because we have all the information cells need to be in a certain organ into a certain function, we can have this as a recipe. And we apply this recipe of those magical cells that I called the stem cell earlier, and we build organ in laboratories. So what is a stem cell? So a stem cell is a cell that has the capacity to replicate itself, to give a sister that is totally similar, a clone to itself, or to give a different cell that is going to give some function in an organ. It can be a blood cells, a nerve cells, or bone cells. So the most powerful stem cells are called the totipotent. They are able to do everything, and the first cells you develop when you have the egg that is formed with the spermatozoid and the oocyte. And in this case, the cells are able to give all the embryo, but also the placenta, all the extra embryonic tissues that are important to develop a baby. But quickly, the cells separate. You have the one making the placenta and you have the one making the embryo. The ones that are inside the embryo, they are a bit less potential. They have less potential. They are called pluripotent, but they can still give all the organs of the body. And then what happened to them when the embryo is growing is that they engage in separate way. So some are going to become the ectoderm, some the mesoderm, and some the endoderm. You imagine 3 different roads they can take. So they restrain again their potential. They become multipotent. And the ones that are ectoderms, they will be able to give the nervous system and the skin. The mesoderm one will be able to give the blood, the bone, and the heart, the cardiovascular system. And the endoderm one are going to give the respiratory tract and the digestive tract. And the body is going to develop, and we are keeping a few stem cells in the organ we need to renew all our life, like the skin or the gut. But those cells, they can give only that tissue after in the adult. So we look to what I explained at the beginning, for the beginning of my career. When we take adult stem cell of the skin, we are able, for example, to rebuild the skin in laboratory. However, we can do only this, which is was very frustrating for scientists. They wanted to have, again, the pluripotent cells that you have in an embryo to be able to do whatever they wanted in the laboratory. And this dream became true at some point, thanks to a Japanese researcher that found a way to reprogram adult cells into pluripotent stem cells, and we call them induced pluripotent stem cells. And this person had the Nobel Prize for this. So Sam can do this.
Yes, yes. So you might have just missed that last bit, but I can put up a poll asking you, what year was the Nobel Prize awarded for this discovery of induced pluripotent stem cells? So let me launch that now. For those that weren't there at the start, we have a way for you guys to all interact with the session, which is these polls. So your options are 2007, 2012, 2020, or 1985. There might have been a clue probably in your career path, Laure, that meant that one option is probably ruled out there. But yeah, have a quick vote on which one you think is likely to be when this when the Nobel Prize was actually awarded for this discovery. Okay, if you haven't put in your answer but would like to do so, please do so. Do so now. Okay. So we've got the majority saying 58% of the room are saying 2012, with the next highest being 2007. Let's have a look and see what people think. Yeah, if you're happy to move over to the next slide, let's see what the correct answer is. Okay, yeah, 2012. So well done for everybody who put 2012. And so, yeah, you're happy to move on now.
So yes, so as I said, they found a recipe. This recipe was to express 4 genes that are listed here: MYC, OX34, SOX2, and KF4. And so the Nobel Prize was given to Shinya Yamanaka and Surgeon Gordon for this discovery, because Surgeon Gordon was the one who showed that normally any adult cells, if you change the program, you replace by the program of an embryonic cells, is able to become again a pluripotent stem cell. So that was an important discovery that he did experiment on frog with this, and he took an adult cells and make a new frog from it. And Yamanaka wrote the recipe that you could do with 4 genes, the same things. So what has happened from there is that a lot of development could happen very fast. because so the first discovery was done on mouse in 2006, and 2007 the recipe was applied to human cells. And people started to be able to do things in laboratory thanks to those cells. And in 2014, we were considering starting to put them back in human to use them in clinic. So that was quite fast. How we take the cells in your body, we can take them like we see in this picture, just taking some few of your cells, and we reprogram them in the laboratory, and we obtain induced pluripotent stem cells from you. What can we do with those cells? We can put them in the laboratory and create some of the organ. So if you have some genetic disease that gives some phenotype, we can after test some drug on those cells in the laboratory. And if we find some drugs that are working, we can give them after to the patient. We can also take the cells and repair them genetic defect and redo some of the part of the organ and engraft again in the human, in yourself, because that's your cells. And as it's very powerful because you do not have immune rejection, because we started with your own cells. So that's a lot of dream around this. So here in the lab, now what we do, if we do this to the recipe to make little piece of skin. And so, like I said earlier, we can do hair follicle. So that can be another poll here to see if you have any ideas, how long does it take?
Yeah, super. So I will launch another poll, which is I'll launch it now. So yeah, how long does it take for skin organoids to develop hair? So have a think about how long do you think it will take for sort of skin organoids grown in the lab to develop hair? So we've got 1 week as an option, 1 month, 3 months, a year. So I'll leave you to have a minute to sort of put in your answers and have a think about what you might think is a rough time scale. So as I say, how long does it take for skin organoids to develop hair? 1 week, 1 month, 3 months, 1 year. Okay, if you would like to submit an answer but haven't done so, please do now. If not, I will close the poll in just 30 seconds or so. Okay, we've got a real spread. So we have 29% saying 1 week, 41% saying 1 month, and 27% saying 3 months, a couple of people saying a year. So what's the right answer?
The answer is 3 months.
Okay, 3 months. Wow.
Yeah, so it's a way, look, if we summarize and you have some little picture of the aspect of those induced pluripotent stem cells and the way we progressively go until something that has some hair follicle. And as you can see, we keep them quite a long time. In culture, we can have the 5 months and do some experiments with them. So here you have a schematic of how the human skin looks. It's a very complex organ, and with hair and sweat gland, muscle, a lot of things. And we do this in vitro nearly perfectly, I have to say. We have sebaceous gland, hair follicle, sweat gland, but it's not totally perfect. And Chloe is going to explain you what we could do better with this.
Thank you so much, Laure, for introducing all of those concepts. So now I'm going to talk about how we're using the prenatal skin atlas that Ferry and Elena introduced in the first Genomics Lite a month ago, and that Laure talked about today, to draw hypotheses. So when we first look at the human prenatal skin atlas, it's a lot of information, a lot of different puzzle pieces you might see that are representing all the different time points that you can see across gestation, all the different donors, the different cell types that we have in the skin atlas. And sometimes it's quite hard to get a global picture of what is happening in the prenatal skin. But thanks to the human Bloodless initiative, we are able to put all of those puzzle pieces back together and to gain a comprehensive understanding of how our human skin developed during the in utero phase. But obviously, this is, even when it's put back together, it's a lot of information that we have. So sometimes it's easier to focus on one piece of the puzzle. So for example, for my PhD, I've decided to focus on macrophages and their role in the prenatal skin. So what are macrophages? They are very fascinating white blood cells that are part of our immune system. And they are very, very important because they are the first line of defense against pathogens in your body. So for example, in the skin, they sit at the barrier between the outside world and the inner world, which is your body. And they are here to prevent you from getting infection and so on. So as you can see here, they have all those little elongation that they use to sense the environment and detect if there is a pathogen or anything that can harm you, and they will eat it up and get rid of it. They're also very important in maintaining a good, healthy tissue because they remove dead cells and prevent them from accumulating and causing more problems if they stay there. Sorry. They're also involved in organizing immune responses. So from looking at the prenatal skin atlas, what we've learned is that macrophages actually seed the human skin very early during in utero life. And what we know is that they do not express any genes that are related to their immune functions. But instead, what we notice is that they're actually expressing genes that are related to blood vessel function. And we also know that in prenatal skin, they are in near proximity to blood vessels. So you have macrophages that are a lot of the time co-localizing with blood vessels. So from everything that we've learned from the prenatal skin, we were able to draw a hypothesis, which was macrophages support blood vessel function during human skin development. And from that, we needed to be able to build a skin model in a dish to be able to validate our hypothesis or invalidate it, obviously. Why do we need a skin model in a dish? They are very important because before we could make organoid in a dish, all of these hypotheses were validated on animal models. So if you take the skin, for example, if you look at the development of the skin of a mouse, it will be very different at the one from a human, obviously, because it has way more hair follicle and we're two different species. So being able to study this development on actual human tissue is a huge advantage for us. And this is why we want to develop those organoids in a dish. We're also able to follow one organoid across the entire development of this organoid. So we have a live happening during the development. But when you look at human sample, it's kind of a snapshot of this person at that time when the sample was taken. So you don't have a live comprehension of the cells. They can also be used as a platform to induce perturbations because, for example, in my project, which is looking at macrophages' role in a human for obvious ethical reason, we would not be able to get rid of all the macrophages of a human and see what happens when we don't have them. But this is something we can do in skin model in a dish. Also, something that is quite important to highlight is that fetal samples are very precious samples and they're quite scarce, and there's a lot of ethical laws around them and how we can use them in the lab. So now that we know why we need them, how can we build a skin model in the lab? So from the prenatal skin, we get a recipe and is apply this recipe and get a skin model in a dish. Obviously, it's not straightforward and requires a lot of hours in the lab, a lot of optimization before we get the end product. As Laure presented, the current recipe we have in the lab for the skin organoids. So here is a picture of a skin organoid actually that we grew in the lab. We generate them from stem cells and we get them with hair follicle, with sebaceous glands, with fatty tissue, some nerve fiber, etc. But this model still has some limitations. So for example, they do not have any immune cells. So for my project that is focusing on macrophages, obviously it's a problem. They also develop very poor blood vessels that are not really functional. So they are actually the perfect model because we can just add macrophages and see what happens to the blood vessels to either validate or invalidate our hypothesis. What we want to do is take the current recipe and make an updated recipe where we have macrophage-bearing skin organoids. So I'll hand over to Sam for the question.
Okay, yeah. So what we're going to ask you is, how long do in vitro macrophages survive? And there's going to be a caveat, and Chloe will highlight what the actual lifespan is in real sort of humans. But yeah, in the lab. So I'm just going to launch the poll now. Someone's already put a question also in the Q&A, which is great. I would encourage everybody else, if they've got any questions, do start putting them in the Q&A bit, and we can get to that later on. So yeah, how long do they survive? 1 day, 2 weeks, 1 month, 7 months? Have a go at guessing which one you think sounds right. Okay, I'll give you 30 more seconds. I'll be kind. 30 more seconds. Oh, and I also forgot to mention when I did the introduction, which feels like a great point to mention whilst you're doing the poll. It's actually DNA Day today. So the 25th of April is DNA Day. So yeah, if any of you joined because of DNA Day, then lovely to have you with us. Okay, I've stalled for enough time, so if anyone wants to quickly put in an answer that they haven't had a chance to do, please do so now. If not, I will end it. Here we go. Okay, so we have got 21% of our audience saying a day, 40% saying 2 weeks, 28% saying a month, and 11% saying 7 months. So what is the correct answer? 2 weeks. Okay, slightly just go into a little bit of detail what the difference is between sort of in-lab and sort of normal in terms of lifespan.
So the macrophages that we're talking about in this poll is the one that we make from stem cells. So once we have them fully differentiated, they can survive on their own for 2 weeks about. But the one that you have in your body, the one that resides in your organs, you will acquire them from birth and, well, in utero life, and they can survive from a couple of years to your entire lifespan. So for example, you have some macrophages in your kidney that you had when you were in utero. So it's quite impressive. So to have our updated recipe, our first step was to generate macrophages in the lab. So as I said, we generate them from stem cells, which are actually the same stem cells as the one that we use to make organoids. And as you can see at the beginning, we make the stem cells aggregate together, and we cook the recipe, we add the ingredient that are needed, and then at the end we get macrophages. So that's our final products, and it's those cells that will live for 2 weeks in a dish on their own. But as you can see, those macrophages are see-through, and it will be quite hard to actually see them once we've added them in the organoid. So then our next trouble was, how can we track them before we add them in the organoid? And that's another poll question for you guys.
Yeah, so what we're going to do is this time I'm going to ask you all to use the chat. Is that we just want you to have a think about how are we potentially going to track them in the lab. So what would we do to them to potentially monitor them and track their sort of their lifespans and stuff like, and monitor them. So put in the chat. So you'll see down at the bottom, we've got the chat button. So you'll feel free to just type in an answer. If you have any ideas, I'll give a couple of, just a minute or so just before we move on. And then we'll go through again. Thank you for those that are putting in their questions into the Q&A button. Okay, so quite a lot of got quite a lot of responses, which is great. Thank you for putting them in. So we've got a few people saying stain them or label with a light reactive gene dye. I've seen a few people say GPS with a laughing face. So yeah, GPS is one of them. Fluorescent gene markers, staining. I think one that says phosphorescence, dye them, quite a lot of staining. So thank you for those that putting in the answers in the chat. What is the correct way? What is the way that you guys do it?
There is different way, and staining is a good answer, but the problem with staining is you would require your sample to be dead, so you wouldn't be able to follow it live. So what we do is we actually make our macrophages fluorescent by using a virus that is non-infectious and then carries, in this case, a little small like red molecule, and it's able to get into the genome of our target cells, so our stem cells, and integrate this molecule inside the genome. So the stem cell then become red or whatever color you want. We decided to go for red. So yeah, quite a few good answer in the poll. So now that we have our macrophages, we made them fluorescent to be able to track them. Our third step is to add the macrophages to our skin organoids. So here we have macrophages and we have skin organoid, and as you can see, they are very different culture that have different needs. So they require different medium. So that's the food we give the cells. They require different culture condition. As we mentioned, they have different lifespan with macrophages that go for 2 weeks, skin organoid that grow for 5 months. So the main challenge here is to be able to combine cells with very different needs. So you might see it as we have our current recipe and we know different factors that are very important in this recipe that you can see them as ingredients. And we want to play around with those ingredients and try to see which is the best combination to get the updated recipe that we want. So this is just a schematic of kind of all the options we would consider. Sometimes it's way bigger. When I did my first experiment, I had 28 different conditions that I was looking at. So it's a lot of work, and it requires thinking of a lot of different ingredients that you could change. But once everything is working, then it's really rewarding. So as you can see in this little video, it's a live tracking. Yeah, it's a live tracking of our macrophages here in red inside the organoids after we added them to the organoid. So the organoid, you can see them as a see-through kind of composition with the macrophages in red on top. So here we are very happy because we have our updated recipe with our organoids that have macrophages inside. But this is not the last step of this recipe, because now we need to make sure that our new model is still faithful to what we see in the fetal skin. So the two main questions are the macrophages also in proximity with blood vessels as we see them in the prenatal skin? And are the blood vessels more functional now that we added macrophages? So here is a reference picture that was taken in fetal skin. And blood vessels in green and the macrophages in yellow here. And this is the network of blood vessels that we are aiming for, because this is the one we see in fetal skin. When we use our old recipe with the skin organoid without macrophages, you can see this very mesh-like, very thick blood vessels that really do not resemble what we see in fetal skin. So this is a very dysfunctional blood vessel network. But when we use our updated recipe, you can see the network starting to resemble really more closely what happens in fetal skin with a thin and very organized network. And what you can also see is the little macrophages in magenta here that are all colocalizing around the blood vessels and doing what they do by supporting the blood vessel growth and organization. And if we take a closer look at our organoids, we can actually see the macrophages here in pink with the blood vessel here in green. And we can see a really close interaction, kind of if the macrophages was hugging the blood vessels. So we were really happy with our and how faithful it was to the prenatal skin. So what we achieved with this updated version of the recipe of skin organoid is a macrophage-bearing skin organoid. We were also able to validate our hypothesis that macrophages are indeed involved in promoting and supporting blood vessel organization. And with this model, we also opened doors for further experiments. So for example, what we could do is look deeper at the mechanisms of how macrophages are actually supporting this blood vessel network. We could also kind of zoom out of the blood vessel and look at the more general role of macrophages during skin development, their interaction with nerves, for example, or the skin cells. And we can also use this platform as a disease model. Skin disease involve macrophages or other you want to add them. For example, eczema and atopic dermatitis, which are two very common skin diseases, are also known to have macrophages that are very pro-inflammatory, but we don't really know the mechanism behind this. So with a model like this, we would be able to study it further. So what I described today was focusing on one recipe and how we used it for my project in the lab. But obviously, the prenatal skin atlas have a lot of different other recipes that are yet to be tried or even discovered, and some are more obvious than others. But this is why this kind of atlas is actually a very, very important resource for us in the lab. And I'll hand to Laure for the final words.
Yes, so what we hope is that we give you hope tonight on what we are doing. And the idea, of course, what you can see in the future is that with all those recipes and those fabulous stem cells, we can rebuild pieces of the body when they need to be replaced because of due to some disease or aging. And so we feel that we grow up inside our tissue culture room, and we hope we transmitted this positive vibe to you. So yes, so thank you very much for your attention. Of course, it's a teamwork doing all this. So we are a large group, like you can see here on the picture. And so a lot of people contribute every day to this effort, some people behind computers, some at the bench with the lab coats, but it's a huge, huge effort. And also we need to thank to all the tissue donors and their family, because without a sample, when you work in human research, you cannot progress. So, and we're happy to take questions.
Amazing. Thank you both for going through that. And I know that there are a few questions already in the Q&A. So if you do have questions, please put them in the Q&A bit for us to get through, and we'll start getting through them shortly. I guess just because you showed that slide last, I guess just to give our audience a little bit of we're kind of a little bit of a forward look, like what do you see this space, especially with stem cells, moving in, say, maybe 5 years or even 10 years? How frequent are we going to be hearing about stem cell research moving forward in the next sort of 5-10 years?
So we think it's going to evolve very fast. The world of organoids is evolving very fast. And in particular, the community also are working on brain cells. They do amazing things. We are now able to do little brain organoid connecting to a computer. And we are going to benefit a lot of AI. AI is helping to refine the recipe extremely fast to have the right cells and also the right treatment to put on them. When I explained that when we have disease, we can have disease modeling in a dish now. We can take, make stem cell from any patient with any disease and try to understand this disease in the dish. So it's going to move very fast in the 5 next year. The problem will be, of course, all the clinical trial to test each time we put cells on the body to make sure, of course, of any consequence that could bring.
Yeah, amazing. Okay, we've got lots of questions popping in, so I'll make sure I go through those before I don't want to hog all the all the questions. So we have one which has said, I'd love to know more about the human models of skin used in the lab. Are these models representative of all skin types, demographics, etc.?
Well, so when you do it from stem cells, like the stem that we use to do the stem cell that we use comes from one donor that is reprogrammed to make the stem cell, the iPS that Laure was talking about, the induced pluripotent stem cell. But a new interest of the lab is to work on what we call chimeroids, which would be using different stem cells, so different donor, to make organoids, so we could include like all the different demographics and so on, all the different plurality that makes that individual, but in a dish. So this is something that is also very important, and the Human Cell Atlas is also focusing on it. We're trying to sample the minority ethnic like backgrounds to make sure that we get everyone included.
Yeah, and I guess it's worth saying that obviously the Human Cell Atlas is a huge global program, right? How many different countries is that again involved? We can share the number on the website shortly, but yeah, it's many global countries involved doing similar work to what you both are doing, right? And sharing that, and that's very much an ethos of where we work in terms of the Sanger Institute, is sharing what we do with the world, and vice versa with other organizations as well. Okay, can genetic technologies such as CRISPR-Cas9 be used to manipulate organoids?
Yes, it's possible. And as I said in one of my slides, we can repair some of the abnormality. And also what we can do is induce a lot of different mutation, and we can tag our cells, and we can put one mutation a cell. And we can see after, when we develop the organoid, if depending, the mutation you put with your CRISPR-Cas9, the consequence it can have. And so, yes, it's something we can use in the organoid.
Amazing. Okay, moving on to the next one is, what makes organoid models for macrophages more important than those analyzed in, say, 2D cultures? So I'm assuming, but if the person who wrote this, if I'm slightly understanding this wrong, I would assume that it's kind of, what's the positive about 3D and 2D when you're looking at these types of things?
So, for example, what is done as 2D usually involves two sites that you would look at. So, for example, if we want to look at the skin, you would have the fibroblasts, you would add your macrophages onto them and see what happens. So obviously you're lacking all the skin and the 3D skin environment with the hair follicle, the glands that you would have, the epidermis in this case. Or you can do another 2D where you would look this time at the keratinocytes, so the epidermis, and add macrophages onto them, and then look what happens. But you're always lacking the environment, and it's something in science that is very important. So when you look at a cell type, you always want to consider the environments that it has, and this is why the organoid is an amazing tool for us, because it reproduces the environment of the human body.
Okay, great. And so I've got a couple of questions that kind of link, and it kind of links, Laure, a little bit to what you were saying about things moving ahead in the next couple of years, which is around AI. So one of those, and so you might have already touched upon these when the question was originally written, but is one, how can stem cells benefit from AI? And then I guess the caveat of that is, how reliable are the results generated from AI or bioinformatics?
So the big advantage of AI is that you can put in the machine as many information as you can find on the way, for example, all the knowledge we have on how the body develops, or anything that can be disruptive, how the disease develop, all this, and the machine is able to integrate all this information and magically to give you insight on the best recipe to treat something. Which drug, we know so many drugs currently that we don't know what they could treat, how they could be useful. And with knowing all the dysregulation or the normal functioning you can have, and have the drug, AI can submit some drug to test in priority, for example. So with the organoid coming from diseased person, those data from AI, we can be faster at developing treatment. So that's the way I think in particular AI is going to benefit to the world of organoid and stem cells. And for the, yes, of course, it's dangerous there can be some danger to use AI. And what is particularly important is the way you feed the machine, the information you give. That will be, of course, critical to what you have on the other side. So that's what is particularly important.
Yeah, and if I can add to that, it's also, this is why we have organoids, to make sure that what we see in AI translates in the dish.
Yeah, okay. So I seen that there is a specific question in the chat which I will not be able to answer, but Laure and Chloe, you might be able to sort of directly answer. So if you're able to maybe write in the chat a response to that question, because I fear that yeah, I won't be able to do it justice. If not, what I can do is I can pass it over to you both via email and get an answer to that person, because it's specific about lab experience and what you do and stuff like that. So we'll look at getting an answer to that more specific one in a sec. So there are a couple more to go through, and the time is flying by. So yeah, thank you for keep putting your questions in. So so someone's put, are you able to alter the epigenome of these organoids to see what effect it has on them? I see that that's triggered a thought-provoking look from the side of
You, no, and yes, I guess it's some of the modifications that can be done, but not the one we are currently doing ourselves.
Okay. Someone's just put, and this might go down to a little bit more into the definition again of induced pluripotent stem cells, but someone's asked, is it possible to create sperm or egg cells from induced pluripotent stem cells?
I think there are some protocol people working on germinal research. I do not say that I have the recipe myself. Something I will have to look in the literature. But lots of cell type we can do now. So, but I'm not sure which levels. We are all very specialized in some field, and the level of you can reach in terms of oversight and the different stage that I would not answer for sure the question.
Okay. This is probably my ignorance coming through now. So we've mentioned obviously these recipe books and that there's different recipes and stuff like that. In terms of for you, how are those actually sort of documented in terms of these recipes, if that makes sense? How do you go and know what is maybe the recipe for this, say, certain organoid or this? Well, like, where is that information and how do you navigate that type of thing, essentially? And forgive me if anyone watching, if that's a really ignorant question up from me.
It's an excellent question, because it's one of the problems the field try to tackle currently, because a lot of people develop their own recipe in their place. So usually you have several ways to make the recipe. You look what happened during the development and you try to copy more or less. Or once we know the gene expression, we try to force the cells to express something. But some of the lab have different recipe, and we know that we are going. It's important because it's like that you have more chance maybe to find the perfect one. So it's not something totally negative, but we will have also to reach a level where some consortium are put in place to validate which protocol is the best one. We call this benchmarking, and particularly for clinic. When we will have some clinical trial or things like that, people will have to use exactly the same recipe to make sure that what they compare will make sense in terms of comparing data made in different laboratory, on different genetic background, on different. So yes, the recipe is as complicated that yes, when you try to look how to make a specific omelette on Google, we have to be careful that there are not too many recipes on the market.
Okay, that's really interesting. I'm glad it wasn't a silly question to ask.
It's not a question at all. No.
So I'm noticing the time, so I will we'll stop there on the questions. I noticed that there is one that came in that was about where you can watch the first part of the this series, this two-parter. I will send it out with the recording of this one. I'll send the recording that happened of the previous one as well, so you get to watch that back. Whilst we still have so many people in the room, which is fantastic, I would like to sort of give a slight teaser, which is towards our summer series. We will be taking a small break from April with Genomics Lite, and we'll be returning in August, and we're going to be taking a slightly different flavor. We're going to be looking at we're going to be hopefully exploring some topics that that are slightly more general in terms of where genomics is interacting maybe a bit more with society right now and stuff like that. But we'd love to hear from our audience as always, and you've been so interactive with us tonight. If there was any topics that you'd love to hear more from our staff like Laure and Chloe on, please do let us know in the chat. I'll leave the chat open for a while, but if you're able to put any topics that you feel that you would really benefit us speaking about and having the expertise from our site talk about, we would be really keen to hear what you have, what any ideas that you have or any topics you want to find out more about. Whilst you're doing that, whilst you're thinking about that, I would like to thank Laure and Chloe for spending their afternoon / going into evening with us. And yeah, it's been an absolute pleasure to have you with us and to hear about all of the stuff that you're working on. It's fascinating, mind-blowing in terms of where it's gone to. I guess so maybe to sort of close off the session, I guess one final question, which is, we kind of had a little bit of a sense from you, Laure, about the steps that it's kind of gone to from where you were starting to where you where it's got to right now. Is what excites you the most looking forward to the next year or so? What are you most excited about moving forward now?
Well, in our case, we want to be able to reproduce some things that happen in the adult also life. We are able to renew our hair. And if we could find a way to have those little organoids doing the same, you know, so, and it's something, because I know several people watching tonight probably wonder if we will be able one day to be able to implant those things. So we want, in our case, is to be able to make, you know, this discovery having more impact on the hair. So each domain has this, you know, like that, looking forward. When I was doing the cardiac regeneration, we were making patch that we could put at the place where people had an infarction. When you have an infarction, all your beating cells are dying and replaced by fibroblasts, and it's why you are in cardiac failure. So researchers want to put patch of cells like that, and so your heart will beat again everywhere. So this will not be in 1 year, but I feel that in 10 years we might be there, and I hope it.
And yeah, Chloe, how about yourself? What's maybe something that still blows your mind to this day about what you're doing, or yeah, what are you most excited about?
What I'm really excited about for my PhD actually is that with the team, we're going to look more into scarless wound healing. So we know when a fetus is scarred, like at the beginning of the pregnancy, like when you wound it, it doesn't scar. But once you are born, obviously you get scar tissue. You can't get grow hair follicle again on the scar tissue, and you get this really ugly part on your skin. And what we would like to study is how you get this scarless tissue that would regenerate completely as it was completely new. And I think that's something that I'm really excited about.
Amazing. Okay, well, once again, thank you so much for spending your afternoon with us, and thank you to everybody who is watching for joining us today. As I said, halfway through DNA Day, so it seems very apt that we've managed to time our Genomics Lite perfectly for that. As I say, we'll be taking a very a short break really in terms of Genomics Lite, and we'll be back again in August for a nice series of events we'll be running one instead of every month. We'll be running them on Thursdays, but each week for a month. So we'll be getting loads of content to you. As I say, I'll keep the room live for a little bit longer, just in case anybody has any topics that they would love us to feature, and we can make sure that we try and cover those in our August session. But if you need to shoot off, please do so. It's been lovely having you with us. And as I say, we'll send out the recording and any links. I know that there was a question about papers and stuff like that to read more. So I will chat with both of you, Laure and Chloe, about potentially anywhere that people could go to find out more. Super. Okay, well, I will, as I say, I'll hang on for a little bit longer, but Laure and Chloe, feel free to shoot off, and anybody else that's on the call. But if you do have any topics that you want to put in the chat, please do.
Thank you very much
so
for having you.
You're very welcome.